Good morning, everyone from Stanford, California. Welcome to the first Plant Cell Atlas workshop, session 2, Tools and Technologies. I'm Zhiyong Wang, the acting director of Carnegie's Plant Biology Department and a member of the organizing committee. So what's the goal of Plant Cell Atlas initiative or this workshop? If we look back at the history of life science research, we would see that one of the constant goals in biology has been to elucidate the structural maps of organisms at increasing resolution, with major progresses made driven by new technologies. For example, botanists and developmental biologists used to study structures of organisms that are visible to the naked eye. Cell biologists focus on everything visible under the microscope. And now we have the map of the genomes. Most of us are now engaged in one way or another in elucidating the functions of genes and the proteins. However, to completely understand how plants grow and function as really complicated self-organizing systems, we need knowledge about what tissues, cell types, and developmental stages each gene is expressed in, where every protein is localized at subcellular level, and what proteins interact with each other in different cell types, and even how metabolites are distributed and transported among different cells and tissues. Such knowledge are accumulating at exponential speeds thanks to the breakthroughs in technology and methodology. Therefore, we think it's time to bring the plant community together, bring our data, our knowledge, and the skills together to build a complete map of plants with molecular resolution. That's the goal, overall goal of this Plant Cell Atlas initiative. And this workshop, the goal of this workshop is kickstart this initiative. So this is the first workshop, and it includes 3 online sessions. We had the first session on vision of Plant Cell Atlas last week, and today, the second session will focus on tools and techniques. Our speakers will present some powerful tools developed in recent years, and we will discuss how these tools can be used broadly and perhaps in your own research, and what new tools we need to develop in order to achieve the vision of Plant Cell Atlas. Now I'm going to turn the mic to Ken Birnbaum to give you a summary of the session 1 and outline of the session 2. Ken.
Okay, thanks, Jim. So this is Ken Birnbaum. I'm one of the organizers tuning in from New York. And so to get you in the right mindset for this session, we thought we would give a visual recap, a sort of graphical summary of the first workshops of the 25 breakout sessions or breakout rooms, and what the conclusions or what the weight of the conversation was. So of course, the first concept that comes to mind is, what is cell identity? And this was a major part of the discussion. What are all the different cell types, and can we define them? But it wasn't just that, it dug a little deeper, and a lot of the discussion had to do with how specific cells are going to respond to the environment. And then in addition, another part of the dynamics of cells, how cells were going to change during development. That is the timing issue. Embedded in these discussions were the idea that perhaps we need to get beyond transcriptomics and start to be able to do this at the cell level, at the protein and the chromatin level. And then of course, the functional questions, which is subcellular localization, protein-protein interactions, things that will help us determine function on a broad scale. Okay, so another big part of the discussion was the evolution question. And so here, can we get beyond our good friend Arabidopsis? That's been a powerful model, but use the technologies at hand to start applying cell-specific information to other models. And a lot of the conversation that came up here was, sure, we'd like everything, but we know we've got limited resource, so where can we function? Where can we focus our efforts? And so specific models came up of where to put that effort, in particular thinking about the species that address some of our major questions, C3, C4 metabolism, symbiosis. Okay, another question that came up quite frequently in the rooms was, what is the sum total of cell-cell communication? That is, we're pretty good, or we're at the cusp of being able to get a lot of information about individual cells, but what about the conversation between them? And then going another step, what about the ways in which we can integrate information at the cell level into the organ or even the whole organism, and how the cells are going to function together? So that's a very broad overview of the discussion that went on among the 25 rooms. And so now we focus on the tools to get there, right? So we're gonna, in a few minutes, after the talks, we're gonna guide you, or we're gonna give you some questions in the breakout rooms. And that's basically want to get you thinking in two areas. What are the high priority current technologies that we could scale up right now to make a useful resource in a plant cell atlas? So it could be some of the ideas that are going to come up. They're going to whet your appetite with some really neat technology, but it could be beyond that. And then another aspect is, what are the moonshots? What are the technologies that we need to develop that will really put a maximal impact on the community and answering some of these big questions? Okay, so just the logistics of what's going to go on, and some technical aspects. So we're going to have 4 lightning talks, 5 minutes each. Again, this is really to whet your appetite and think about the technologies we can use. We're going to hold questions after individual speakers and go to a Q&A session that'll last 15 minutes. So as a result, you'll be able to type your questions into the chat box, but please put in parentheses or in the beginning who you want to direct that question to. Now that can be to all the speakers as well, but be specific about that. Okay, so the breakout sessions will follow. This is the really important part of the workshop. So that's going to last about 45 minutes. If you don't want to attend a breakout session, please leave and come back. It just helps us with logistics. Take our word. And then once the breakout sessions open, just wait a few minutes. We've got to put the individual moderators in by hand. You can start introducing yourselves or talk about the weather or something. Okay, at 9:30 then, we're going to come back and we're going to try to summarize all our discussion. And that'll be the last session. And then a couple of reminders. It's spring, so if you want to tweet, there's our handle. And one favor, except for the speakers and organizers, could you keep your video stream off to conserve bandwidth? I guess its rule is bandwidth over beauty. So at that, I will turn it back to Zhiyong, and he's going to give a quick introduction of our 4 speakers. So take it away, Zhiyong.
All right, so today we have 4 speakers. The first speaker is Zhender Jones, who is actually Carnegie alumni now at Cambridge University. The second speaker, Tess Brennan, is a postdoc at UC Berkeley, and she did her PhD at Alice Ting's lab at Stanford. And the third speaker is Markita Lange from UC Berkeley. And the fourth speaker is Becky Bart from Danforth's Plant Science Center. Zhender, take it over.
All right, so good morning, everyone. Thanks for joining. I'm Markita Landry. I'm an assistant professor in the Department of Chemical and Biomolecular Engineering at UC Berkeley. And today I thought I'd take a bit of a zoomed out view. I have, I think, 1 slide of our lab's data, but to bring into the conversation the use of abiotic materials, specifically nanomaterials, for genetic manipulation in plants, though certainly these materials have also been used for the delivery of nutrients and pesticides at a rapidly growing pace in agricultural biology. So the motivating factor for a lot of the work that we do, especially for genetic modification of plants, is in the projected dearth of nutrient-rich food, especially in the light of population growth and a decrease in the availability of arable land mass. So specifically, projections estimate that food production must increase by 70% in the coming decades so that we can feed our growing global population. Now, genetic engineering of plants is one of many possible ways of addressing the potential shortcomings that we're about to face in our food supply. And so within the context of what's currently being done in genetic engineering of plants, the tools that exist for introducing foreign DNA, RNA, and proteins, which are necessary for the genetic manipulation, are twofold. And they're depicted here, the first of which is a biotic method, so specifically agrobacterium-mediated delivery, in which we take advantage of a pathogen-plant interaction, in which agrobacterium can be genetically manipulated at the bench to contain a gene of interest that can then be transfected into plant cells, integrated into the plant host genome, and producing transgenic plant tissue that can then be regenerated into transgenic plants. Agrobacterium-mediated delivery suffers from drawbacks of species range limitations. So some species, especially wild species and even subcultivars of the same species, might not be susceptible to agro-mediated infection. Conversely, biolistic particle delivery is an abiotic method whereby DNA, RNA, or protein can be dehydrated onto the surface of a gold particle and can be shot into plant tissue using a high-pressure gene gun. So the advantage here is that we have a method that allows for cargo-independent delivery, whereas agro is largely confined to DNA delivery alone. The gene gun method, however, usually suffers from low transformation efficiency and cell damage due to the biolistic nature of the delivery, which can pose some challenges upon regeneration. So with both of these methods, there's a lack of control, both over the frequency of transgene integration, especially for biolistic delivery, and also the location, which could potentially disrupt endogenous genes through integration of the transgene or other parts of the plasmid in the plant host genome. So these drawbacks aside, genetic modification of plants has generated still a lot of success. The United States is one of the global leaders in the production of GM crops, which are listed here. And a nice international example that I like to give is the development of golden rice, in which the gene pathway for the production of vitamin A was synthetically, genetically reconstituted into rice, so that this essential vitamin could be endogenous to a transgenic strain and then disseminated to communities for which this vitamin was found to be deficient. Now, again, despite the advances that we've made in generated GM crops, today the average GM crop still requires about 13 years of R&D at a cost of nearly $140 million, 40 of which are regulatory. So there's a significant time and also financial burden with going through a GM regulatory pipeline, where at least in the United States, GM crops are classically defined as those that have been generated through biotic or pathogen-based methods for which there is integrated foreign DNA into the host genome. So part of what my lab works on is in developing nanoscale tools that are able to efficiently deliver DNA, RNA, and hopefully in the future proteins as well in an efficient manner, in an abiotic manner that would be plant species independent and ideally tissue independent as well. And lastly, one that has greater control over the integrating nature of the plasmid, so non-pathogenic and non-integrating, with an eye on the regulatory side of things.
1 minute left.
And so here, I'm showing some recent results for both the delivery of DNA, which allows us to express a foreign transgene across multiple plant species. And conversely, a separate platform in which we can deliver small interfering RNA into plant tissues to knock down, in this case, a transgene, but also endogenous genes. And part of the features that we've observed for both platforms is in the transience of the expression or of the gene silencing, in a way that we can also confirm a lack of transgene integration, such that when we combine these tools with CRISPR-based delivery, for example, of plasmids or of the RNP itself in the future, we'd be able to genetically edit a plant, perhaps avoiding transgene segregation that is currently necessary with other tools. Lastly, to get a sense of how the regulatory map is moving within this space, GMOs have been differentially regulated across the globe for decades, ever since they became a part of our food supply in the 1980s, and how non-transgenic genome edited plants using CRISPR technologies and others will be regulated, are also starting to show heterogeneous patterns of adoption depending on local regulatory bodies. So in combination with the use and increased use of nanotechnology and plants in agriculture, I think these are also important conversations to have. So lastly, just a brief acknowledgement of the lab. Guzda Demer did most of the work that I've shown today. Also, Huan Zhang, who's a postdoc in the lab, and I look forward to our discussions in the breakout sessions.